Hello, everyone. I'm sorry I could not join you in person. My name is Petra Sušjan. I work at the National Institute of Chemistry in Ljubljana in Slovenia. And in our group, we are, in our smaller group within our lab, we are trying to come up with RNA targeting strategies. And particularly today, I would like to focus on one of these strategies, namely the antisense oligonucleotides and their potential in treating the syndrome. So let's start with the basics. So we cannot go past the central molecular biology dogma to understand RNA processing. So our genes are encoded on the DNA, and each gene corresponds to a protein. However, there is an intermediate molecule called the RNA, that is the transcriptional level. And there's a lot happening in this molecule, this single-stranded molecule, before it is cleared off to be able to code for provide the coding sequence for the protein. Namely, in its precursor form, the pre-messenger RNA, it consists of both coding regions called exons, as well as the intermediate regions called introns. And one of the main things that is happening in the process of RNA maturation is that a big multi-protein complex called spliceosome removes these introns to join the coding region into a frame region coding a specific protein. In addition, there are other mechanisms that are taking place. For example, something called alternative splicing. So at the point where these introns are excised, it's not necessary that exons step together in this sequence. Certain exons can be skipped. So in the end, we can also get, in addition to the full-length RNA, also its splice variants with a different sequence of exons. And in addition to this, there's also fast capping and polyadenylation tail addition to the microRNA to really make sure that it is not degraded and stable to act as a matrix for the translation. And so this strategy, knowing how this occurs in nature, almost invites that we think about how to exploit this knowledge for therapeutic targeting. And with the advance of the so-called antisense oligonucleotides, we are able to do so very easily, I would say. So antisense oligonucleotides are single-stranded, nuclease-resistant synthetic oligonucleotides that bind to the complementary region. Full complementarity needs to be achieved on the target RNA, and thereby in different mechanisms, with different mechanisms, alter its processing and expression. And yeah, those are relatively short, so like 15 or 12 to 13 nucleotides. And simply this DNA RNA duplex leads to degradation. So we have to make ASO nuclease resistant, and we do that with various chemical modifications. So the chemical modifications were developed in 3 different generations. So the earliest one is phosphatidylate modification, where the oxygens were replaced with the sulfur in the phosphate group. However, this one is slightly tricky because while it enhances the stability of the ASO, it also interferes with affinity. So the second generation, for example, was the addition of methoxyethyl moiety, and this actually helped with retaining the high affinity of the ASO. Then the third generation was the phosphorodiamidate morpholino. This one combines both high affinity, high nuclease resistance, and also it circumvents some of the disadvantages of the previous modifications, namely it does not have a huge protein binding ability, which is a problem with the other modifications, because we want the ASO specifically to bind to RNA, not to proteins. So the mechanisms of action of ASOs vary. Here are just the three most common ones. For example, ASOs can cause RNA degradation through the action of the RNase H enzyme, and this leads basically to degradation of RNA and downregulating protein expression. It can cause steric hindrance, which basically means that we mask important motifs within the RNA, thereby making the RNA inaccessible to translational machinery. It can lead to the arrest of 5' capping. This also leads to downregulated protein expression. But then one form of steric hindrance is also splicing modulation. And in this way, some naturally spliceosome will attach to the exon intron junctions and it will splice out the intron. However, if we cleverly position the ASO on these junctions, we are able to induce either exon skipping or or exon exclusion, depending on what splicing motifs we are targeting. And this can then lead to altered protein, either upregulated or downregulated. So this is the timeline of ASO approval. You can see that for the neurodevelopmental illness, several ASOs have already been approved. So the first ASO approved was Omidiersen. And this one had quite a nasty application route. It was into the eye. So we have since moved on from this kind of applications, specifically the eteplirsen and nusinersen. This was a big milestone in the ASO development because these two are one of the most, these two marked the beginning of use of ASO for treatment of neurodevelopmental diseases. But eteplirsen not extremely efficient, so new varieties of ASOs still continue to be developed and approved. For example, Goldirsen, Vitorarsen, Casimersen. One that I will also briefly mention is Milasen. This is a first personalized ASO that's been used only on one patient. And there are, of course, also several ongoing clinical trials for Dravet syndrome, Angelman syndrome, and so on. So I'll just briefly focus on the mechanism of eteplirsen, which is used to treat Duchenne muscular dystrophy. So in this disease, what is specific that there are these hotspots for exon deletion that leads to loss of function in dystrophy. However, what's important to note that not all exons are completely indispensable for this protein function, and therefore some of them, it was found out, can be actually deleted, and this would lead to a shorter dystrophin that would retain part of its function and would therefore result in a milder form of the disease. And that's exactly what eteplirsen is trying to achieve. So this is used in patients that, for example, have a deletion of these two exons, 49 to 50. And then this deletion, of course, changes the, makes a frame shift in the coding sequence. And therefore, it was figured out that if an additional exon is actually deleted, then the reading frame would be restored, thereby leading to this milder form of the disease. That's one way of approaching disease treatment with ASO. Another one is nusinersen, which is used to treat spinal muscular atrophy. in this disease, the mutations in the SMN1 genes are the one that leads to loss of its function. However, here they did not target this gene that contains the mutation. They instead went to its homologue, SMN2 gene, which had in healthy individuals a problem that it had a strong intronal splice silencer, which caused the deletion of exon 7 and production of a poorly expressing SMN2 variant. Now, what they did with nusinersen, they are blocking this splicing silencer, thereby causing inclusion of the exon and a fully functional SMN2 transcript, which is used to replace the missing function of the SMN1 gene. And then I would also like to mention Nielsen. This was quite an inspirational ASO. The story dates back to 2019, where they developed it at the Boston Children's Hospital. And they were approached by the family of this young patient called Mila, who battles the Batten's disease, which is the disease has problems in toxic waste removal from the lysosomes. And what they figure out when they sequenced Mila's genome, they figure out that in between her exon 6 and exon 7, a transposon or so-called jumping gene was inserted, which is quite low odds for something like this happening, because this happens all the time in our genome, but it usually targets the non-coding regions and thereby has no influence. But with her, it was different. The inclusion of this retrovirus caused an alternative exon to appear, and once this alternative exon was spliced, it led to premature stop codon, which of course led to an unfunctional protein. And what they did is mask this alternative splice site and restored the normal splicing. So it was considered that this drug that was approved in an accelerated process really extended her life for a few years, but she eventually passed away because the disease, once they started treating, was already in a quite advanced stage. But well, how can we, based on all these promising results, utilize ASO in CTNNB1 syndrome? So it's been explained in previous talks that CTNNB1 gene codes for beta-catenin. CTNNB1 gene, just like all genes, comes in two forms, two alleles, each of them inherited from each of our parents. And specifically with this gene, both of these alleles need to produce beta-catenin in order for it to be sufficient to exercise its roles. And this beta-catenin famously has two roles. Actually, they are quite opposing. First one is more structural. It's part of the EC-adherent complexes, where it conveys cell-to-cell adhesion, connections of cytoskeletons of neighboring cells. And the other one is a role in regulation of YNT signaling pathway, namely it acts as a co-transcriptional factor with the TCF, LDF transcription factors, and it stimulates the transcription of YNT target genes. Now, in CTNNB1 syndrome, there is a mutation in one of these two alleles, which means that at worst it causes loss of function, loss of half of the beta-catenin. So it means that the roles of beta-catenin are, of course, downregulated, resulting in a pathological phenotype. How can we utilize the knowledge of the biochemistry behind the beta-catenin function to implement our antisense oligonucleotides? So we have to know, and it has been discussed in previous talks, that in the absence of the YNT signal, beta-catenin is continuously degraded. Namely, it is bound by this so-called destruction complex, whose role is to phosphorylate it and lead to its ubiquitination, and ultimately it is degraded on proteasome. So herein lies the therapeutic opportunity. We can allow the accumulation of beta-catenin by trying to find ways of inhibiting degradation. And this is exactly what we are trying to achieve with our ASOs. We have designed several ASO candidates targeting the YNT signaling pathway, and then we test the effect of the ASOs on beta-catenin activity with various reporter systems. This is just one of them, and then we check the effect of YNT transcriptional program. And we have got a few promising candidates. However, one particularly stood out. So we were able to obtain a dose-dependent increase in beta-catenin activity with one of our candidate ASOs. So the way activity of beta-catenin is measured in this assay is by utilizing a so-called top flash assay, where if beta-catenin is functional, it interacts with the transcription factors and binds to the promoter. However, in our reporter system, this promoter regulates the by luciferase, which has a very easily measurable readout, namely the luminescence. And in the non-functional beta-catenin, this readout will be absent. And in addition to obtaining a concentration-dependent increase in the activity of beta-catenin, we also checked the YNT target genes. We found a significant fold induction, so we treated the cells with ASO in several of the YNT target genes. So does Antisense make sense? There are pros and there are cons, as with everything. Only the time will tell which in the specific setting of CTN and syndrome outweigh each other. However, I would emphasize that with the ASOs, we do not interfere with the endogenous regulation of the genes, so we leave the promoter regions in the DNA intact. We are addressing the transcriptional level. And in addition, the modification is transient. There is no permanent modification, which is important, that should some adverse effects occur, we can very easily cease the treatment. It's easier to regulate dosage, and should there be need, drug can also be tailored to the person specific mutation. There are some cons. The synthesis is quite expensive. There's limited options for rational design. It means that many candidates need to be tested before we find a successful one. There are some delivery challenges with ASOs, but also new delivery techniques are approaching, so this could turn actually into an advantage. And there are also some adverse effects associated with certain types of chemical modifications. So with that, I would like to acknowledge other people who are working on this particular project with me, especially PhD student Mateja Maruna. And also I would like to acknowledge the funding agency. We obtained funding after very selective process, so we are very motivated to get better results. Thank you very much.